Welcome everyone. Today we'll explore proximate analysis, a fundamental set of tests used in food science, agriculture, and feed industries to determine the basic chemical composition of any organic material. Proximate analysis gives us a nutritional snapshot. It tells us the major components, not the detailed chemistry, but the big picture. It's been used for over 150 years and is still the gold standard for nutritional labeling and feed formulation. Proximate analysis is used everywhere, from your breakfast cereal to livestock feed to new product development. It ensures quality, safety, and transparency for consumers and regulators alike. Proximate analysis breaks down any food or feed sample into 6 key components, and this slide shows all of them at a glance. Let's go through each one. First, moisture. This measures the water content of the sample. It's done by oven drying at 100 to 105 degrees Celsius until the sample stops losing weight. Simple, but essential. Second, ash. Ash measures the total mineral content, the inorganic matter. We burn the sample in a muffle furnace at around 550 degrees Celsius. Everything organic burns off, leaving behind minerals like calcium, potassium, and phosphorus. Third, crude protein. This is a calculated value. We measure the total nitrogen in the sample using the Kjeldahl or Dumas method, then multiply by 6.25. Why 6.25? Because proteins are roughly 16% nitrogen on average. It's called crude because it assumes all nitrogen comes from protein, which isn't always true. Fourth, crude fat, or ether extract. This measures the lipids and oils using a Soxhlet extractor. The sample is continuously washed with a solvent like petroleum ether, which dissolves the fat. The solvent is then evaporated, and the fat is weighed. Fifth, crude fiber. This measures the indigestible plant material, things like cellulose, hemicellulose, and lignin. We digest the sample with dilute acid and then dilute alkali. What's left is the crude fiber. Sixth, carbohydrates. Unlike the others, carbohydrates are not directly measured. They are calculated by difference using this formula. 100 minus the sum of moisture, ash, protein, fat, and fiber. This fraction includes starches, sugars, and some soluble fibers. It's a rough estimate, and it accumulates any errors from the other tests. And that's it, 6 components, 6 methods, 1 complete picture of what's in your sample. Don't confuse proximate analysis with ultimate analysis. Proximate is for food and feed. It tells you nutritional composition. Ultimate is for fuels. It tells you elemental composition for combustion calculations. Different industries, different tests. Proximate analysis is used across many industries. From the cereal you eat for breakfast to the feed your livestock eats to the research lab developing the next superfood, it's everywhere. Proximate analysis is a fantastic tool for getting a quick snapshot of what's in a food or feed sample. But it's not perfect. In fact, it has several important limitations. Let's go through 6 of the most common challenges you need to be aware of. First, crude protein overestimates. The Kjeldahl method measures total nitrogen, not just the nitrogen that comes from protein. If your sample contains non-protein nitrogen sources like urea or ammonia, they get counted as protein too. This means you can get a false high protein reading. In animal feed, this is a real issue. Urea is sometimes added, and it inflates the crude protein value. Second, crude fat may under or overestimate. The Soxhlet extraction method uses a solvent like petroleum ether to extract lipids. But not all lipids are equally soluble. Some polar lipids, like phospholipids, may not be fully extracted, so you might underestimate fat. On the other hand, other non-fat compounds like waxes or pigments can be extracted too, leading to overestimation. So take crude fat values with a grain of salt. Third, crude fiber underestimates. The acid and alkali digestion used to measure crude fiber is quite harsh. It dissolves some of the fiber components, particularly certain hemicelluloses and soluble fibers. So the crude fiber value is often lower than the true total dietary fiber. This is why modern food labels now use more specific dietary fiber methods. Fourth, carbohydrates accumulate errors. Remember, carbohydrates are not directly measured. They are calculated by difference using this formula. 100 minus the sum of moisture, ash, protein, fat, and fiber. This means any error in any of those 5 tests ends up in the carbohydrate value. So if your protein reading is too high, your carbohydrate reading will be too low, and vice versa. Fifth, limited compositional detail. Proximate analysis tells you about broad categories, but it doesn't tell you the specifics. For example, it doesn't tell you which sugars are present: glucose, fructose, sucrose. It doesn't tell you the types of starch or the profile of dietary fiber. It doesn't identify individual minerals like calcium, potassium, or magnesium. It just gives you total ash. If you need that level of detail, you'll need more advanced techniques like HPLC or ICP-MS. Sixth, it's time-consuming. Some of these tests take a long time. For example, the crude fiber digestion takes several hours. Soxhlet extraction for fat can run for 6 hours or more. And some moisture and ash methods require overnight drying or heating. For a busy quality control lab with hundreds of samples, this can be a major bottleneck. So to summarize, proximate analysis is a great starting point. It's simple, affordable, and gives you a quick nutritional snapshot. But it's not a precise tool. It gives you estimates, not exact values. And it won't give you the detailed chemical fingerprint of your sample. For routine screening, it's perfect. But if you need accurate, detailed data, you'll need to complement it with modern analytical methods. So, modern analytical techniques can fill in the gaps. They give us more accurate, detailed, and targeted data. Let's look at 6 modern alternatives that are transforming food and feed analysis. First, direct sugar measurement using HPLC. High-performance liquid chromatography can separate and measure individual sugars, like glucose, fructose, and sucrose, with incredible precision. Unlike the carbohydrates by difference approach, this tells you exactly which sugars are present and in what amounts. This is critical for nutrition labeling and managing sugar content in products. Second, specific starch analysis using enzymatic methods. Instead of lumping all carbohydrates together, these methods use enzymes to break down starch into glucose, which is then measured. This gives you a true starch content, not just a rough estimate. Very useful for cereal products, processed foods, and animal feed. Unlike crude fiber, which underestimates fiber, the AOAC method captures both soluble and insoluble fiber. This is the method used on modern food labels. It gives you the true total dietary fiber, much more accurate and useful for consumers. Fourth, mineral profiling using ICP-MS. Proximate analysis gives you total ash, but it doesn't tell you which minerals are present. ICP-MS, inductively coupled plasma mass spectrometry, can measure individual elements, like calcium, potassium, magnesium, iron, and zinc, at very low concentrations. Fifth, near-infrared spectroscopy, or NIRS. This is a game changer. NIRS uses light to analyze a sample without destroying it. No chemicals, no lengthy digestions. It's rapid, results in seconds. And it can predict multiple components at once: moisture, protein, fat, fiber, and even individual sugars. It's widely used in the grain industry, dairy processing, and feed mills for real-time quality control. This is essential for both human nutrition and animal feed formulation. Sixth, amino acid analysis. Crude protein tells you the total nitrogen content, but it doesn't tell you the quality of that protein. Amino acid analysis breaks down the protein into its individual building blocks. This tells you which amino acids are present and in what amounts. This is critical for assessing protein quality, especially in infant formula, sports nutrition, and animal feed where specific amino acid profiles matter. So where does that leave us? The answer is, when you need accurate, detailed, and specific data, these modern methods are the way to go. HPLC for sugars, enzymatic methods for starch, AOAC for fiber, ICP-MS for minerals, NIRS for rapid screening, and amino acid analysis for protein quality. Choose the right tool for the job, and you'll get better data, better products, and better outcomes. To wrap it up, proximate analysis is your go-to method for understanding the basic composition of any food or feed. It's simple, affordable, and gives you the big picture. Just remember its limitations, and use modern tools when you need more detail. Thank you so much for watching. If you found this video helpful, please hit that like button. It really helps the channel. Subscribe and turn on the notification bell so you never miss our upcoming videos on food science, chemistry, and analytical techniques. And drop a comment below. Let me know which topic you'd like me to cover next. Thanks again, and I'll see you in the next one. Bye for now.